Good evening, ladies and gentlemen. I welcome you all for this our monthly clinical meeting. As I told you from last month, we are having live webcast of this program, and this could be reviewed anytime once you visit the same site. Just to keep you informed that in the continuing dental education programs that we have planned for this weekend, that is on Sunday, there is a provincial clinical meeting that will be held in Batticaloa. 4 lectures been lined up. If you happen to come that way during the long weekend for holiday, maybe you can visit and attend the lecture and the fellowship that's going to be afterwards. And in January, second week, again, we are having the regional scientific session in Jaffna. The Jaffna branch will be organizing that. I'm informing you all in advance. Those who like to join us can inform our office and attend that conference. At this point, I would like to invite our chairman of our continuing dental education program, Dr. Thutikaran, to introduce the speaker today.
Thank you very much, sir. I have the real honor of introducing Dr. George Chantadias, one of our own product, who is well ahead in dentistry in the international world, well known in the international arena. It's a real honor and privilege to have you today, sir, at this event. Dr. George Chantadias completed his BDS in 1977 from the University of Peradeniya, and he completed his MS in 1985, and he is a oromaxillofacial surgeon, and he is the first oromaxillofacial surgeon Sri Lanka produced. I mean, after the MS was formed and OMF category was there, he is the first person to be produced from the faculty. And he did his PhD in 2003 in functional anatomy of TMJ. And presently he is the associate professor in clinical anatomy at the University of Otago, New Zealand. And his special interests are on biomaterial in orthopedic applications. It's not related to dentistry, it's in orthopedic applications, biomaterials. And also he is interested in clinical anatomy of craniofacial region and facial reconstruction as well. So he is an anatomist as well as a clinician. He has so far published about 50 scientific journals, and he has 3 international patency on biomaterials. And he has supervised over 10 PhD students and more than 10 master students. So it's our honor to have you here, sir. I would invite you to the podium to give you the lecture. Thank you.
Thank you very much for those kind words. I'm humbled, maybe I don't deserve all the praise, but yeah, it's a privilege to come before you, and I must thank you, President, for inviting me, and also thank you for coming and listening to me. So some of these logos that you see here, I'm attached to the anatomy department at the University of Otago. So this is the logo for University of Otago. That's the logo for the anatomy department. And also I'm a member of the clinical anatomy research group. which I lead at the moment. And also I'm also in the management committee of this Otago Center for Bioengineering and Nanomedicine. It's a multidisciplinary organization. People from all different disciplines in the university are in this center. We are working on bioengineering, and in fact there's one student who's planning to come and do a PhD over there. So this is the building the Center for Bioengineering is housed. So we got some laboratories and offices here. So that's about the first slide. So I'm going to talk to you about new directions in endodontic therapy. So when you look at endodontics, nothing much has changed since I did endodontics. That was in the previous century, and for decades. Of course, there are some new materials have come, but the basic principles the same. Okay, nothing has changed. Now, but when you look at the modern thinking in treatments, people are going away from replacing diseased tissues with artificial stuff. There was a time everything artificial, like we used all kinds of things, stainless steel, chromium, cobalt, titanium was the fashion. But now, recently there was a decision was taken by the number of groups like College of Surgeons, etc. Anything you put in, if it stays on after the job is done, even titanium, it becomes a foreign body and prone for problems. People thought that titanium was the silver bullet, that all bone related problems will be solved with titanium. Initially, yes, it was fantastic, it worked perfectly. But now the oldest titanium implants are coming to about 15-20 years. Now we are seeing the problems. Titanium has wear particles. You find the titanium particles in lymph nodes, livers, kidneys, all over the place. Okay, so that's why this decision has been taken. We must not leave stuff inside people if you can help it. So the new thinking is you must replace diseased or lost tissue with the same tissue. Okay, if possible. That's where bioengineering comes in. Okay, so what we are trying to do is replace a disease or lost bone, lost anything with the same tissue, or get the tissue to regenerate. So we have been working in other areas in bone, but we thought about why don't we take it to the tooth. So in the tooth, the live stuff is in the pulp and the dentine. So that's why endodontics came in. So what we are trying to do is we are trying to regenerate the dead pulp and the dead dentine so that the tooth can heal itself. So that is this work we are doing. That's the essence, and I'll take you through what we have been doing. Right, the down arrow, no? Yeah. So the background to this work we have published in one of the editor of this journal is one of our own, Lakshman Samaranayaka in Hong Kong. He's the editor of this journal, and so we published this work. These are some of my students and myself in advances in regeneration of dental pulp. So it appeared this year in this particular journal. So that's the background to this work. If you're interested, you could. Right. So I'm not going to dwell here because all of you are dental surgeons, so you know all about this. And so anyway, so due to infections, trauma, the tooth can become non-vital. The pulp dies. Okay, some of the dentine get infected. So when that happens, only treatment available to us is the root canal therapy. So, and I'm not trying to, this is like showing your grandmother suck eggs. I'm just basically, this is what, so the treatment involves opening up the pulp chamber, remove the disease, remove the necrotic pulp, remove the infected dentine with mechanical means. You use chemicals to flush the stuff out, straighten the canals, then put sealers in, seal the canals, and then you restore the crown. So this is essentially the endodontics principles are endodontics, and this has not changed for the last, I think, 50 years. Now don't get me wrong, there's nothing wrong with this, because it's an extremely successful treatment. Now see, the success rate is about between 78 to 98%. I mean, so you can ask, if it's so successful, why try to fix something which is not broken? That's very true. Why are we trying to mucking around here if this is so successful? Although endodontic treatment is very successful, long-term studies have shown this endodontically treated teeth, the loss of endodontically treated teeth in the long term is quite high. You get recurrent caries, fractures, and all that. And also, it's a dead tooth. Okay, it will not function. You will get the of course, to a degree, you get the proprioception because the periodontal membrane ligament, but you don't have any other sensations. Temperature, the pressure, etc., etc. And so, because of those problems, and also you have put a lot of synthetic material into the body. So these are all foreign bodies. Okay, so the modern thing is you must try and get rid of those foreign bodies, because they may eventually cause problems. So that's where we are looking at alternative treatment for dead teeth, looking at a biological approach. Now when you look at biological approaches in tissue engineering, you got to have these three factors. You got very important that you have a scaffold. A scaffold is a network, or like a skeleton. Now even the human body, everything is hung on the skeleton. You got to have some kind of a framework to hang stuff on. So to do that, you need a scaffold. Okay, I'll come to the scaffold later. You have all kinds of scaffold. So you need a scaffold, and then you also need stem cells. Because not having a scaffold is not enough. It's just the framework. You got to have the guys who do the job. The stem cells should get in, and then they should differentiate into different types of cells, and those cells will lay down the tissues, whatever the tissues required for that particular region. And also you need the growth factors, which are secreted by the cells, okay, to recruit the local and the general mechanisms. Of course, if you get these two right, that will look after itself. So now these are the conditions that you need if you want to regenerate any tissue, including pulp. Now, Dr. Amuludama asked me to say a few words about the current state of affairs in New Zealand in endodontics. So I just spoke to Robert Love, he's the professor of endodontics over there, just get an idea what they are using. Now we can see there's nothing different what you do here compared to what they do over there. The stuff are the same. So the main antimicrobial irrigant that they use during chemo-mechanical canal preparation is 2.5% sodium hypochlorite. I'm sure that's what you use too. Okay. You can use chlorhexidine 0.2%, but it's not as good as sodium hypochlorite, because the chlorhexidine do not dissolve the necrotic tissue, whereas the hypochlorite does it. Okay. So then they also use a chelating agent. So you know what a chelating agent is? It removes the calcium ions, weaken the hard tissue so that it can be easily removed. So they routinely use a chelating agent, the EDTA, 17%, okay, during the canal preparation to dissolve and remove the inorganic dentine debris here. So once the canal is prepared, okay, then the canal is dressed with calcium hydroxide. Just tell me if you are not doing anything different here. Calcium hydroxide, because the pH of calcium hydroxide is about 12, quite high, highly alkaline, extremely good disinfectant. So leave that dressing for 1 week. That reliably gets rid of the infection, get the canals ready for root filling. And finally, root filling is done using cements and sealers, GP, and the sealers are, there are many sealers that they use, but most commonly I was told by Robert Thomas that's the Densplice H plus that they use. It's an epoxy resin based cement. Do you all use this stuff? Yes, because he says that it's epoxy resin is very sticky and it seals the canals, etc. well. And also, sometimes they use use MTA, mineral trioxide aggregate, MTA cement, in place of GP and other sealants. You put the only MTA to seal the whole thing. Okay, so that's another. Do you all use MTA here? I'm sure.
Not so much.
Okay. It's good that
because of the cost.
this is very costly. That's very strange because MTA, you know, MTA is purified Portland cement. That's exactly what it is. It is just Portland cement purified. That's MTA. It's a cement that sits in a aqueous solution. So you can buy a bag of cement and do the endodontist. I'm sure it will work if you get rid of the acidity and all that. That's what is MTA. That's where these people make money. You know, a bag of Portland cement will produce huge amount of MTA. So that's what I'm trying to show is even the so-called fancy material which comes to the market is nothing fancy for this bucket chemistry practically. Okay, so these are the reasons that we want to move on to the next stage of treatment for especially for endodontics. Right now, when you look at if you want to use tissue engineering for endodontics, you can do it two ways. You can have the dead tooth and you can get the pulp to regenerate within the dead tooth. That's one approach. Okay, you can do that. Other approach is grow the pulp in the laboratory. You can do that, get a scaffold, grow the pulp, pulp-like tissue, and then implant that into the dead tooth. Okay, so those are the two approaches you can take. And we are
Can I
looking
ask
at
you a question there?
Yeah.
What happens if the pulp is
Infected.
Infected, yeah.
Yeah, so you have to get rid of the infection by just standard methods, really.
First.
Yeah. Then we first you have to get rid of the infection, then you can regenerate the pulp and make the tooth alive again.
Is that the foreign bodies, are not perfect.
Yeah. The tissue
No,
engineering.
my question is, if you're going to get rid of the foreign body that we should get through,
Yeah.
you go through almost all the stages.
Not yeah.
Can we have the questions after
Yeah.
because
Yeah. No, the thing is, you don't have to do the ethical stuff. You don't have to worry about straightening the canals. You don't have to do anything. Just put some antibiotics even, get rid of the infection, and then it's ready for regeneration. You don't have to prepare all these perfect canals and all reaming and all that. It's not necessary. Right. So these are the two approaches that you can take. Right. Now, stem cells. I said that you need three things. You need scaffold. You need stem cells. Few words about stem cells because I know it's an interesting subject. So there are two types of stem cells. There are the embryonic stem cells. Then you have the adult or the postnatal stem cells. The postnatal adult stem cells, there are two types. Hematopoietic based stem cells. These are the stem cells that you get from blood, available in blood. You can extract from blood, including the cord blood. Okay, and then you have the mesenchymal stem cells. These stem cells are found in bone marrow, in fat, adipose tissue, including the pulp around the tooth. You get stem cells. So those are the mesenchymal stem cells. These are the two different types of stem cells. That's my cursor. Embryonic and the adult stem cells. Adult ones, two types: hematopoietic blood or the mesenchymal stem cells. When you look at the differentiation ability of the stem cells, you can divide the stem cells into four groups. You have the totipotent stem cells. Now these stem cells, one of these cells can produce a whole individual. Okay. Now these stem cells are found in the early embryos, in 1 to 3 days embryo. You remember your embryology goes to 2, 4, 6, 16, 30. At that stage, if you take one of the cells out, this totipotent, you can produce a whole embryo. That's how you do cloning. Okay. Then we have the pluripotent stem cells. Now these stem cells can produce any cell in the body. Therefore, can produce any tissue in the body. Now these pluripotent stem cells are found in late embryos between 5 to 14 days later on, or these pluripotent cells are also found in the cord blood. Okay, I was talking to you Chief Justice recently about, in fact, in Sri Lanka we are exporting placenta systems. I said it's like taking your babies. You should keep and extract the stem cells from these placentas rather than exporting them somebody else somewhere else in the world are doing it, because it has very good quality stem cells. So I may write a short report to that we should have a cord blood storing facility in Sri Lanka. So in the future, in next generation, we'll have stem cells when they come to our age. Right. So the pluripotent stem cells, those cells can differentiate into any cell in the body and produce any tissue. Then we have the third type, the multipotent stem cells. Now these cells can differentiate into certain cells, therefore produce certain tissues. So the stem cells which you can obtain from pulp are multipotent cells. Okay, pulp, bone marrow, fat, all those are multipotent cells. They are okay, they are good, but they are not as versatile as the pluripotent cells. Then finally you have the unipotent cells. They can differentiate only to one type of cell. For example, the stem cell found in skin. They can become only skin cells, nothing else. Right. So talking about the pulp-derived stem cells, we said it's a multipotent mesenchymal stem cell. So you can get, we are talking about stem cells obtained from the pulp. But don't forget, there are stem cells in the periosteum, there are stem cells in the apex, there are stem cells in the exfoliated teeth, milk teeth. You can extract stem cells. Okay, people have done that work, but for us, we found this the most convenient place. Go for the pulp. And these pulp-derived stem cells, of course, can regenerate into dentine pulp-like complex. Okay, these stem cells can produce dentine and pulp, not the enamel, because it comes from epithelium. Also, these stem cells are neurogenic. They can form new nerve tissue. So you can use these pulp-derived stem cells to regenerate, say, spinal cord injuries. Somehow, I don't know, but it's possible because they can turn into nerve cells. You can these stem cells, pulp stem cells, can turn into myogenic, can produce muscle. I don't know, maybe heart muscle. These are the possibilities for the future. They are adipogenic. They can produce fibroadipose tissue. After mastectomies, maybe you can regenerate the breast tissue. Okay, with these stem cells. All these come from the pulp. Doesn't mean that you can use only on teeth. You can use in lots. They are also chondrogenic. Cartilage, maybe you can use on the joints. So these are the possibilities for the future. If you have a good, satisfactory or efficient way to extract these stem cells, that's what we've done. So this is how we did it. First of all, you got to have proper laboratory. It's called the physical containment level 2, PC2. My lab is PC2. So PC2 lab has the necessary equipment. You got to have laminar flow hoods, So air is blown out through the hood so that no contaminants will go in. You will not infect your material. You got to have water bars. There's a whole swag of stuff that you should have in a PC2 lab. You have white coats, gloves, etc. Okay. So you got to have the proper laboratory. This is not fancy stuff. Just common sense aseptic technique. Then I got my staff trained in cell culture. The students and the staff was going to do this work. They had the idea. They have grown other cells, but I got them to grow these two cell lines. Now these are called cell lines because these are immortalized cells. They don't die. These L929s are fibroblasts. They are fibroblast-like cells. Originally, in the 1970s, it has come from a fibrosarcoma of one particular rat, okay, somewhere in the United States. Now these cells are found in most labs. So the good thing is, I have these cells. Somebody else in Michigan will have those same cells. So if I do an experiment, somebody can repeat it in Michigan because they also have the same cells, L929. That's why we call the L929. And also, I got them trained using these particular cells, the MDPC23. These are not available commonly. This is only available, that's how the Michigan came to my mind. Only in Michigan, University of Michigan, one of the researchers there has produced this cell line, which is odontoplasty cell line. Okay, odontoplasty is so I had Robert, my colleague, had contacts with them, and we managed to get a vial down, FedExed it, grew the cells. First better we propagated, and then one of the idiots in my lab has allowed the liquid nitrogen to run dry, and all the cells died, and so I had to contact them again and managed to get another second vial. It's kept like gold, and we propagated immediately. Now the cells are kept in several vials, so if one goes bad, we still have them. So the MDP cells are again immortalized odontoblasts, the cells which produce dentine. So the staff, they practice growing these cells also. Now talking about immortalized cells, these cells are cancer cells. These cells do not die, they keep propagating. And also, today we have the techniques to convert a normally differentiated cell into an immortalized cell. You can make a cell immortalized. And because we are now understanding the apoptosis, all cells in the body are programmed to die. They know when to die. They know how many should go through certain number of generations, and they should die. It's all now, in the past we didn't really understand the mechanism, but now we do. So that's why it's very important for cancer therapy and all that, understanding apoptosis. So anyway, to culture, so we got trained culturing these cells, and also, yeah, that's all right. You can see me. And also, we practice extracting pulp from pig teeth. Pig molars are very similar to humans. So that's the preparation, and then we got human ethics approval because we are going to use human teeth. It's not a long process, quite a tedious process to get the human ethics. We got the approval, that number, and we got 20 wisdom teeth, freshly removed wisdom teeth from the dental school, from the oral surgery. Luckily, I have my colleague, not one, two there, and Dr. Associate Professor Rohana De Silva provided us with the teeth. So he used to contact my student, and he used to rush and carry the physiological solution, immediately pop the tooth into the solution, bring it down, and take the pulp out. And so I show you some images. These are the L929 cells in culture, looking through an inverted microscope. That's how they look like. They look like typical fibroblasts, the spindle shape. If you remember your histology from your BDS, these are fibroblasts, okay? They keep growing. These are the MDPC23 cells. They are slightly different. They are the odontoblast cells, okay? And so the pig jaw, here we practice to remove pig pulp from a pig molar tooth. And now in a nutshell, this is how we extracted the stem cells. Okay, I should take you through this, and then I'll show you the images. So we got the wisdom teeth, then we extracted the pulp from the wisdom tooth, human wisdom tooth, under aseptic conditions, and then we broke the matrix of the pulp. Okay, we have to disintegrate the pulp matrix to release the cells. So you use enzymes to release the cells, and then we grew the cells. We cultured the cells, and then we identified the colonies of stem cells. We purified them looking at the colonies of stem cells, and then there is another technique called flow cytometry. I will not go into detail, but with flow cytometry, you can purify these cells further. You can get very pure preparation of stem cells by doing that. So once we got those stem cells, we confirmed that these are really stem cells by looking at the proteins which are found on the surface of the cells, okay, by immunocytochemistry. And also we did scan electron microscopy to see that they are definitely stem cells. We also did another thing. We converted some of these cells, we differentiated these cells into odontoblasts. Okay, you can do that. Okay, I'll comment there. And then once we differentiated, we want to see that will these cells lay down dentine? Okay, so we grew the cells after converting them into odontoblasts. We grew the cell in culture, and then we looked if they are laying down dentine. Alkaline phosphatase enzyme, so we checked whether alkaline phosphatase level is going up. We looked whether the proteins which are found in dentine, are they present there? Is these cell cultures getting mineralized? We look for that. And also we looked at the histology. Does it look like dentine? Okay, and so I'll show you the results now. So, in fact, my student prepared all these slides. You've done a good job. Right, that's the advantage of being a leader, principal investigator. You have lots of people to do the donkey work. Anyway, so we had this is the one of the human wisdom teeth. So we cut the tooth at the amylocemental junction, open the pulp chamber, extracted the pulp into Hank's balance. This is a physiological solution. pH, the osmotic pressure is the same with all the inorganic salts which are found in the blood. And then we use enzymes, collagenase and dispase, these to break down the matrix, pulp matrix, to release the cells, wash the cells, centrifuge the cells, okay, and then we put the cells to a cell strainer. Size of the pores, if anybody interested, is 70 microns, so that only the stem cells will go through, any cell clumps will be removed, and then resuspended the cells, and then we have this equipment in our lab, a cell counter. It automatically, in the past you had to use a hemocytometer, put a microscope and count cells. Now you just put it in, it gives you the count. Count the cells to make sure, to find out the cell density. After that, we cultured the cells. We grew the cells in a carbon dioxide incubator. This is a standard incubator, 5% carbon dioxide, 37 degrees centigrade, the temperature, 100% humidity. Okay, so when we grew the cells, we could see the cell colonies. We isolated the cell colonies. Then we took these cell colonies through 3 to 4 passages. A passage means a generation. Okay, you grow the cells until they completely cover the bottom of the flask. Okay, about 80 to 90%. We call it confluence, and then you break the cells up with trypsin, regrow them. So that's a generation. Okay, so we took them through 3 to 4 generations. It takes about that amount of passages to cells to be. Initially they are too immature. So some of the images. Now this is how the cells look. The stem cells taken from pulp looks like in day 5 in the cell culture. Okay. By day 7, can you see these cells going into forming colonies. Okay, so these are the stem cell colonies. By day 12, you can see very distinct colonies. So when you see a colony like this, immediately we put a cylinder, glass we call a floating cylinder. Put a cylinder around that colony and isolate the colony from the surrounding cells to prevent them getting contaminated. Okay, so after like that, we propagate the cells and then we store them in minus 80 degrees freezer for future work. I'll go through quickly these are the techniques I'm telling you how we did the thing. Then we use this the technique called the flow cytometry. What this technique does is, every cell has proteins on the surface. So stem cells has certain proteins. We looked at these. We took these 3 proteins. Okay, these are called surface markers, and we use these proteins to identify the cells and separate the cells from the other cells. That's what we did using in this technique. Okay, so we use these surface markers to purify the cells further, so that we have a pure preparation of stem cells. Okay. Don't worry too much about these stuff. These are signals. So in flow cytometry, we found there were 2 populations of the stem cells. There's a smaller cell population, a large cell population, but they were the same, no problem. But this, for the future, we'll keep this in our mind. There are maybe 2 slightly different population of stem cells. That's how a pulp-derived stem cell looked like under scanning electron microscope. And also when you zoom in, you can see these processes put out by the stem cells. Okay, so this is how the stem cells look like under the microscope. How did we confirm that these are stem cells? We use immunocytochemistry. So what we did was we get antibodies against those surface proteins, antibody, and to the antibody a label, a dye is attached, a fluorescent dye. So when you put the antibodies onto the cells, if those are stem cells, those antibodies will get attached. So we can see an image showing the cells are glowing green because so we can confirm that these are the the stem cells. We look for all three surface markers. Then we converted the stem cells into odontoblasts. Okay. So when we check the alkaline phosphatase production, you can see from day 7 onwards, day 14, day 21, 28, alkaline phosphatase levels are going through the ceiling. Okay, so these are the stem cells have changed into odontoblasts. The stem cells that were not changed are not producing any alkaline phosphatase. These are the controls. Okay, so we know they have differentiated into odontoblasts. We did the Van Giesen staining. Van Giesen is a normal light microscopy. These pink areas indicate collagen. So these cells are not only become odontoblasts, they are producing collagen. So that's another thing odontoblasts do. So by finding collagen, we know they have differentiated into odontoblasts. One cause of staining, the black patches indicates mineralization. The dentin is not only turning into dentin, the dentin is becoming a hard tissue. Okay, so we compare to the control, you can see mineralization occurring. So now we know our stem cells have become odontoblasts and they are laying down dentin. There's another method, alizarin red. These blobs, red blobs, indicate areas of calcification. So another confirmation. We also look for proteins found in the dentin matrix. Dentin sialoprotein, dentin matrix protein 1. So these immunocytochemistry, the brown staining indicate those proteins are found. So if something looks like a duck, quack like a duck, it must be a duck, isn't it? So we got all this information. So now we know the stem cells became odontoblasts and they are laying down dentin. So we nailed that. Okay, now we are happy. We got a technique. In fact, to do all this work, we required only pulse from 1 to. In fact, we can produce unlimited amount of stem cells. So that is where the versatility is. You know, we can produce bucket dose of stem cells, and not only for teeth, maybe for other stuff. I don't think anybody has done this before. So these are highly good source of stem cells. If you can get a bit of pulp, you can have unlimited supply of stem cells, pulped stem cells. Right. So that's the stem cell part. While that was going on, we were also, stem cells is only just one link in that triad. We also needed a scaffold. So we were going to do, we started working on producing a scaffold based on sheep wool. Okay, this is one of my students did it. I don't have the artistic ability to make slides like this. Anyway, so scaffold, to be used for any tissue engineering, a scaffold had to be biocompatible. It should not kill the cells. It should allow the cells to attach and proliferate. And also the scaffold in this game, once it has done the job, it should slowly wither away and die and disappear. Okay, it should break down. It should not produce any toxic byproducts. It should disappear biologically. So that's another feature that you should have. It should have some physical properties. Okay, it should be able to hold its shape. And also for endodontic purposes, this is where the biggest hurdle was. This scaffold should be able to be pushed through a narrow gauge needle. Okay, and once it goes through the needle, when it comes out from the other side of the needle, it should become a gel again. Okay, and that's what we had a lot of problems to solve that. You could produce gels, but when you push through a needle, it becomes a liquid. Okay, so it took us about a year to develop this scaffold. And also when you look at the literature, there are a few other groups working. I know there's a group in Hong Kong. There may be Germans also definitely must be working on these things. And also Hong Kong and mainland China. But they are all working on collagen. If you do we did a lit search. They are all working on collagen-based scaffolds. Collagen is very easy to use. And there's a lot of knowledge in using collagen. But I do not touch collagen. Do I have time to? Maybe some other time. The reason is collagen comes from animal carcasses. Okay, the collagen for the whole world comes from New Zealand and Australia. These are the only two countries which is prion free. The day we find prions in New Zealand herd of sheep or cattle, that's the end of the collagen market. Okay, all this collagen stuff it's not when it's not will happen. It will happen. It's only matter when. okay, because collagen is animal-derived collagen. Anyways, that's why the reason I'm working on using keratin. So keratin, when you look at a sheep wool fiber, is 95% protein. It has different types of protein, but it has this intermediate filament. This is one of the fractions. Is a structural protein. Okay, so we extracted this intermediate filament protein. Now, when you look at the keratin as a biomaterial, the journey, the whole thing started in 1995 when a Japanese scientist Yamauchi, not met him, I hope to meet him one of these days. When he published a paper on, he developed a method of solubilizing wool and efficiently extracting keratin. We started working around 2000 on this area, but we didn't publish anything around until about 2010, because during that time we were capturing the intellectual property, because there was a lot of marketing potential. In fact, now some of the there are 3 products we are already in the market based on the work that we did. To show you the patents, we got 2 patents on this. There's an international patent, US patent. You can see yours truly is one of the inventors, and so we nailed it using keratin for orthopedic applications, and we sold the university sold the intellectual property to this company. Now it's owned by Keraplast Limited. They are in San Diego, Texas. That's a multi-billion-dollar company. Okay, they are now marketing it. We also published about 4 papers on keratin as a biomaterial in decent journals. These are quite first top 5 to 10% journals in biomaterials, and also we produced about 5 confidential industry reports, and we got paid about $250,000 for this work. So I have a track record. My team has a track record on keratin. That's why we use keratin. Keratin is a very versatile biopolymer. Once you solubilize keratin, it's an extremely versatile biopolymer. You can control the polymerization. You can go from a gel by lightly polymerizing. You can produce transparent membranes. You can produce fibers. In fact, we developed a resorbable suture. Highly polymerized state, you can produce screws and plates for bone fractures. Okay, this is where the clinical trials are going on at the moment. So these are examples. We did a number of large animal studies where we implanted the keratin scaffold in long bones of sheep, in the tibia of sheep. That's the original bone. This is the keratin scaffold. You can see the bone is growing into the scaffold. That's called osteoconduction. That happens with lots of other material. But see this bone, the red color is bone. That's keratin. You can see the bone forming separate from the periphery. That's osteoinduction. Now this happens only with your own bone, autologous bone grafts. So we found that our material has the same potential as an autologous bone graft. That's why lots of people became interested. Now you can see here in a close-up, that's keratin, that's bone. See, keratin is being converted into bone. And also the beauty of keratin as scaffold is collagen. If you put collagen into a bone lesion, after a week the collagen is gone. Okay, macrophages remove the collagen. Of course, the bone will fall, but that newly forming bone has no relation, no resemblance to the original collagen scaffold. Whereas the keratin stays on and acts as a template. It guides the bone formation. It's really guided bone formation. So you can have a certain shaped keratin scaffold, and after 8 weeks, the keratin will be gone. There'll be, instead of keratin, there'll be new bone, exactly the same shape. Okay, so that's the beauty of this material. That's why those biotech companies were very interested. So this is the work I've done on bone. Maybe, Asal, if you invite me again, I'll come and talk to you about that stuff. I thought I had spoken about it in the past. Anyway, so our hypothesis was, if it's going to work for bone, it should work for dentin, because dentin is very similar to bone. That's why we thought of using keratin to produce the scaffold for regeneration. So now I'm coming to the end of my talk. So it has 3 stages. So first stage was preparing, extracting the intermediate filament protein successfully and producing the hydrogel. It took about a year to complete that stage. Second stage is, once the gel was produced, we have to see whether the gel has cytocompatible, biocompatible. We grew the different types of cells, L929s, MDPC23s, odontoblasts, to see the cells will happily grow in this scaffold. We have done that. successful. The stage 3 is the in vivo study with Kamal last. We are coming to that very soon. So, show you some this thing. So, once we extracted the protein, we did an electrophoresis. You know, heard of Western blotting. So these are the protein bands. They are the molecular weights between about 45 to 60 kilodaltons. So we know this is intermediate filament protein. A 2D gel, this is more difficult to do, but a 2D gel also shows us that it is the molecular weight is right, and these are definitely type 1 and type 2 intermediate filament protein. So we got a technique to extract the intermediate filament protein successfully from a concoction of wool proteins. Then once we got the protein, we formulated about 100 different gels. Okay, we tested all different sizes and shapes. We use a rheostat. This is a machine you can check the gelation properties. After doing all that, we selected one formulation, which to show that, unfortunately, there's a little video. Maybe it doesn't work. Anyway, this is a 21 gauge needle. Okay, the gel is in the syringe. When you push through, it will go through the syringe. When it comes out, it turns into a gel again. Okay, so now we know it will maintain, and we check using a rheostat whether there's any difference between the gel which comes out through the needle and the original gel. No, it's the same. Okay, so we think we have cracked that. Okay, so we got the gel, and the gel can be pushed through a fine gauge needle. It will come through the other end and become a gel again. And also we freeze dried the gel, looked under scanning electron microscope to see how well the protein are interlinked. We are happy with the way. So this is how it looks. Okay, this is how even contact lenses look if you freeze dry and look it under a microscope. Okay, so these are the protein septa with large amount of spaces for water. We are happy with the construction, and also cytocompatibility. We grew the cells, and say after 24 hours, 48 hours, the cell numbers have almost doubled, and we use the assay called the live dead cell assay. The live cells appear green, fluorescent green. If they have any dead cells, they would fluoresce red. Now you can see there are no red cells, so all the cells are good. They are live and they are growing well, so we know it's cytocompatible. And then the final stage, we are going to do an in vivo study using rats, Sprague-Dawley rats. I want to use some other breed of rat called the Lewis rat. They are genetic clones, but their teeth are too small. We had to use loops of number 3 magnification to do this. And so this upper jaw, first and the second molars, we have open the pulp chambers, and we are going to implant the gel with or without stem cells, then seal it, and keep it for 4 weeks, 8 weeks, and see the regeneration of pulp. We have started the pre-pilot study. These results will be available in another 3 months. Okay, so that's the work we have done. We developed the stem cells, we developed the scaffold, and the significance of this research is we will be able, with this work, to find a method to regenerate pulp and dentin in the dead tooth, so that you don't have to stick the all kind of foreign bodies into teeth, and the tooth will heal itself. And to do this work, we got about $35,000 from the New Zealand Dental Research Foundation, Maurice Piker's Trust, $5,000, and the Anatomy Department supported with about $15,000. And finally, I have to thank these people. That's Robert Love, the professor of endodontics. Azam Ali he's from Department of Applied Science. He's a polymer scientist, so we are collaborating with him to develop the scaffold. You know, Dr. Rohan De Silva, he provided with the wisdom teeth. Lavanya is my PhD student who's working on scaffold. Isaac Kang, he's an honor student working on the stem cells. Russell is my postdoc fellow working in my lab. And finally this guy, he's 6 foot 8 inches. He had to duck, this guy, he had to duck his head when he goes. He's my lab assistant, so very tall guy. He doesn't like to be called tall. And that's all. And if the chair agrees, I'm happy to answer any questions.